Phase 2b in clinical trials is best understood as a decision point. At this stage, a treatment has usually shown an acceptable safety profile and some early signal of activity. The question now is whether that signal is strong enough at the right dose and in the right patient population to justify moving into phase 3. This is what makes phase 2b different from earlier exploratory work. Phase 2a may still be looking broadly at activity, dose range, or early assumptions, while phase 2b is more focused. It usually tests a smaller number of viable regimens and produces evidence that a development team can act on. The chosen treatment approach is tested more deliberately. That includes the dose, endpoint, study population, comparison group, and analysis approach that may later inform phase 3 planning. This is why phase 2b is sometimes described as a mini confirmatory study because its role is to reduce uncertainty before a much larger investment. Design choices also carry more weight at this stage. Randomized controlled comparisons are commonly used because they give a clearer basis for estimating treatment effect. Blinding may also be important, especially where endpoints depend on clinical judgment or patient-reported information. Here, endpoint choice becomes especially important. The endpoint needs to reflect a meaningful treatment effect and support the next development decision. Depending on the indication, this may be a clinical outcome, a validated biomarker, or a composite endpoint. Operational detail also matters. Visit timing, dosing records, rescue medication rules, lab uploads, query resolution, and endpoint adjudication can all affect interpretation. In earlier exploratory studies, a slightly wider visit window might be acceptable. Timing matters more in this phase because the study is trying to estimate treatment effect at defined time points. Here, the study population often starts to look closer to the intended later stage population. Inclusion criteria is clearer, comparisons are tighter, and the protocol usually leaves less room for interpretation. Phase 2b can also expose assumptions that would cause problems later. Eligibility criteria may create too much heterogeneity. Or an endpoint may be clinically relevant but difficult to collect consistently. Stratification factors may also need refinement. These findings can help teams strengthen the study model before phase 3. Statistical planning depends heavily on this evidence. The effect size and variability observed in phase 2b often feed into phase 3 sample size calculations. If those estimates are unstable, overly optimistic, or affected by avoidable operational noise, the next trial can be poorly calibrated from the start. Common problems include slow recruitment, high visit burden, missing data at key time points, too many treatment arms, too many secondary objectives, and too much flexibility in the analysis plan. These issues can leave sponsors with data but no clear answers. An inconclusive result is harder to use because it leaves the team deciding whether to spend more time and money answering a question that might have been answered more clearly the first time. There is no fixed shape or timeline for phase 2b. Duration depends on the indication, endpoint, follow-up period, and recruitment feasibility. Really, the study should be planned around the evidence needed for the next decision. While the term phase 2c is sometimes used, it is not a standard label with a consistent meaning. Clearer language is usually more useful. Teams should state whether the study is refining dose, supporting endpoint selection, or enabling phase 3 planning. Ultimately, the role of phase 2b is to give teams a clearer view of efficacy and safety at a viable regimen while exposing operational issues that could become harder to fix in phase 3. To learn how Quanticate can support your next trial, email inquiries@quanticate.com or request a consultation through our website. Thanks for watching, and don't forget to subscribe for more practical insights on clinical trials and modern drug development.